Students, hope you are well, staying at home safely in this corona period. Today, I am going to talk about the second week of development. Of course, it is embryonic development. Our objectives are, we will discuss about the day 8, 9th, 11th, 12th and 13 days development. Second week of development is also known as week of 2. This is the first week of development. We have seen in our previous class, and this is the figure of the uterine tube, ovary and uterus, and all this structure shows the events occurring in the first week of development. Here we see this is an ovary and this is the Graafian follicle. From here the secondary oocyte is ovulated and it is entered into the fallopian tube. Here we see the transport of sperm. Then this is the time of DNA duplication. This is the zygote formation. After zygote formation, we know that there is a cleavage division and that this is the 2-cell stage of the zygote. There is 4-cell stage of the zygote, 8-cell stage of the zygote, and ultimately this is the morula formation on the 3rd day of the intrauterine life. And this morula ultimately enters into the uterine cavity. And on around the 5th day, morula forms blastocyst. Blastocyst is an important structure. It composed of 3 parts: inner cell mass, outer cell mass, and the cavity, blastocell cavity. And these blastocysts, around the 5th to 6th day, implanted into the uterine endometrium, which is known as implantation. All these events occurs during the first week of development. This is the picture of the zygote, 2-cell stage, 4-cell stage and the morula stage. And here the picture of the blastocyst. This is the inner cell mass, this is the outer cell mass, and this is the blastocyst cavity. This is the figure of uterine wall. Here we see this is the layer of endometrium and the outermost thin layer is perimetrium. This endometrial layer, again we know that, is divided into 3 layers: stratum compactum, stratum spongiosum and stratum basale. First 2 layers, stratum compactum and spongiosum, is known as functional layer. Here we see this is the functional layer of the endometrium where our implantation takes place, and this is the stratum basale. Now we start the second week development. First of all, at the day 8, here the important events are embedding, tropoblast, development of tropoblast, embryoblast, cavities, and what are the changes occurring into the uterine stroma. All these topics will be discussed for the day 8. This is the day 8 blastocyst stage of the embryo. This is the uterine stroma. This is the uterine lining epithelium, endometrial stroma. This is our blastocyst. At the 8th day of development, blastocyst is partially embedded in the uterine endometrium. The tropoblast, which is the outer cell mass, is differentiated into 2 layers: outer multinucleated syncytiotropoblast and inner single layer of cytotropoblastic layer. This one is the cytotrophoblast, and this one is this deep green color is the syncytiotrophoblast. This is multinucleated cellular layer which has no cell boundaries, and this cytotrophoblast is the single nucleated cell layer. These both are the derivatives of outer cell mass. Now what is the derivatives of the inner cell mass? Inner cell mass also differentiated at the day 8 into two important cell layers. First of all, this is the columnar blue color cell is known as epiblast, and the lower yellow color cuboidal cell is the hypoblast. Both these layers form the bilaminar germ disc, which is the important event that occurs during the second week of development. So the inner cell mass give rise to the epiblast, this columnar cell, and this hypoblast, the cuboidal cell. They form a disc-like structure, so they are known as the bilaminar germ discs. Meanwhile, within this epiblast cell, a small cavity appears. This cavity is known as amniotic cavity, and the cells that forms the roof of the amniotic cavity comes from epiblast, and now it is known as amnioblast. So these are the amnioblast, these are the epiblast, and the cavity between them is known as amniotic cavity. And the hypoblast is also facing towards the cavity, it is known as blastocyst cavity. Now comes to the endometrial stroma. These endometrial stroma become edematous and highly vascular following the implantation, and they are loaded, and the cells are loaded with glycogen and mucus. So there are four important events occurs during the 8th day. These are the partial embedding of the blastocyst, formation of the cytotrophoblast, syncytiotrophoblast, formation of epiblast, hypoblast, formation of amniotic cavity, and the endometrial changes. Now come to the events that occurs during the 9th day of intrauterine life. There are also four important events, that is the embedding, lacunar stage, Hoscher's membrane, and primitive yolk sac. On the day 9th, the blastocyst is more deeply embedded in the endometrium, and the penetration defect, and this penetration defect is closed by the fibrin coagulum. So this is the syncytial layer, this is the cytotropoblastic layer, and this is the epiblast, this is hypoblast, this is amniotic cavity, and this is our blastocyst cavity. What happens during the 9th day? Here, first of all, in the syncytial layer, some vacuoles appear here, and these vacuoles gradually become fuse with each other and form lacuna. Now these spaces, white spaces, is known as lacuna, and the tropoblastic stage at this stage is known as lacunar stage. One important point you have to know that this is where the germ disc is present. This is known as the embryonic pole, and the opposite side is known as the abembryonic pole. Now formation of Hoscher's membrane or exocoelomic membrane. How it is formed? Some cells from the hypoblast migrate inside the syncytial layer and form the Hoscher's membrane. So again I am telling, Hoscher's membrane is formed by the cells derived from the hypoblast. This yellow color thin membrane is the Hoscher's membrane, which lines the inner surface of the cytotropoblast. This is the cytotropoblast, and the inner lining is known as the Hoscher's membrane or exocoelomic membrane. When this membrane is formed, the blastocyst cavity is now known as the primitive yolk sac. So, at the day 9th, we got the lacunar stage of the tropoblast, Hoscher's membrane, and the primitive yolk sac. Now come to the day 11th and 12th. Here, embedding, it's an important point. There are also formation of uteroplacental circulation, extraembryonic mesoderm, and formation of chorionic cavity. This is the picture of the day 11th and 12th blastocyst stage of the embryo at the day 11th and 12th. At this stage, the blastocyst completely embedded in the endometrial stroma, and the surface epithelium almost entirely covers the original defect in the endometrial wall. This is the surface defect, completely covers the surface epithelium, completely covers the defect. Now come to the formation establishment of uteroplacental circulation. Here we see the syncytial cell erode the maternal blood vessels. Here we see the syncytial cells erode the maternal blood vessels. So the maternal blood enters into the lacuna of the syncytial lacuna, and blood, so maternal blood fills up the lacuna, and in this way uteroplacental circulation is established. I am telling again, the syncytial cell erode the, here we see the syncytial cell erode the maternal sinusoids. So sinusoids and sinusoidal blood enters into the lacuna. When the maternal blood begins to flow into the lacuna, the uteroplacental circulation is established. This is the one point. Second point is formation of extraembryonic mesoderm. Here we see a new space in between the primitive yolk sac and the cytotropoblastic cell. This loose connective tissue comes from the hypoblast, and this is known as, this loose connective tissue layer is known as extraembryonic mesoderm. Where this layer comes from, cells of this layer comes from, this comes from the lining of the primitive yolk sac. So the lining of the primitive yolk sac produces a new layer which is known as extraembryonic mesoderm in between the primitive yolk sac and the cytotropoblast. Soon cavities appear in this mesodermal cell layer, and when these cavities fused with each other, they form a cavity. This is known as extraembryonic coelom or chorionic cavity. So in between these extraembryonic mesoderm, large cavities will soon appear, and it is known as the chorionic cavity or extraembryonic cavity. This mesoderm is divided into two layers. One is somatopleuric layer, another one is splachnopleuric layer. Here we see that primitive yolk sac is covered by a red colored layer. This lining which surrounds the primitive yolk sac is known as splachnopleuric layer, and The mesodermal layer, which covers the inner surface of the cytotrophoblast as well as the amniotic cavity. This is known as the somatopleuric layer of extraembryonic mesoderm. So, we have learned that the extraembryonic mesoderm has two layers. One is placnopleuric layer, another one is somatopleuric layer. Now come to the another important point, that is decidual reaction. What is this? After implantation, the endometrial stroma cell become polyhedral and loaded with glycogen and lipids, and also the intercellular spaces are filled with extravasated fluids, so the tissue become highly edematous, and all these changes occurs in the endometrial stroma is known as decidual reaction. It is an important part of the uterine endometrium after implantation. Again, I am repeating the important events that occurs during the day 11th and 12th are the establishment of very important uteroplacental circulation, formation of extraembryonic mesoderm, chorionic cavity, and the decidual reaction. Now day 13th of the intrauterine life. Here the important events are formation of primary villi, secondary yolk sac, chorionic cavity, chorionic plate, and connecting stalk. This is the figure of the day 13th blastocyst. Here we see that the surface defect at the uterine surface epithelium is completely healed up. This is our syncytial layer. This is cytotrophoblastic cell layer. This is the amniotic cavity. This is bilaminar germ disc. This red color lining inside the cytotrophoblast is the somatopleuric layer of extraembryonic mesoderm, and this red color covering lining our previous primitive yolk sac, it is the placnopleuric layer of extraembryonic mesoderm. Now come to the formation of primary villi. How it is formed? Here we see the cytotrophoblastic cell. These cell columns proliferate and gradually penetrate the syncytium. So this cellular column with its covering syncytial layer is now known as the primary villi. This is the primary villi. This cellular column with covering syncytial layer is known as the primary villi. So these are the primary villi. Now the formation of secondary yolk sac. How it is formed? Hypoblast cell. This hypoblast cell produces some additional cells that migrate along the exocoelomic membrane. This cell proliferate and gradually forms this secondary yolk sac. That is, the hypoblast produces some additional cells that is forms along the exocoelomic membrane and produce a secondary yolk sac. So secondary yolk sac is formed within the primary yolk sac. What will happen the rest of the primary yolk sac? The larger portion of the primary yolk sac which remains in this area pinched off, and it may remain as an exocoelomic cyst due to formation of a large chorionic cavity. This is the remnant of the primitive or primary yolk sac as a exocoelomic cyst in the large chorionic cavity. What is the chorionic plate? This somatopleuric layer of extraembryonic mesoderm that lines the cytotrophoblast forms now the chorionic plate. This is now known as the chorionic plate, and this is the chorionic cavity. What is this? This is the connecting stalk. The part of extraembryonic mesoderm connects these bilaminar germ discs and the amniotic cavity, primitive secondary yolk sac cavity to the trophoblast by the portion of the extraembryonic mesoderm that is known as the connecting stalk, which will later on become as an umbilical cord. So this is the day 13th development. Again I am repeating: formation of primary villi, formation of secondary yolk sac, then formation of chorionic cavity, chorionic plate, exocoelomic cyst, and the connecting stalk. Now come to the abnormal implantation. We already know that implantation takes place in the anterior or posterior wall of the uterus close to fundus. If implantation takes place outside the uterus, it results in an extrauterine pregnancy or ectopic pregnancy. What are the sites? These abnormal implantation sites are the ampullary region of the uterine tube, tubal pregnancy, intestinal implantation, implantation to the internal os, ovarian implantation, and also the abdominal cavity implantation. And another important ectopic pregnancy implantation site is the pouch of Douglas. This is the pouch of Douglas. It is an important site of ectopic pregnancy, which lies between the posterior surface of the uterus and the rectum. There is a space which is known as rectouterine pouch or pouch of Douglas. We have already told you that the second week of development is known as week of 2 because it has some important structures, that is, all are formed in two layers of embryoblast, epiblast and hypoblast, 2 layers of trophoblast which is known as syncytiotrophoblast and cytotrophoblast. 2 membranes are formed here. One is Hoscher's membrane, another is extraembryonic mesoderm. 2 sac is produced here, primitive yolk sac, secondary yolk sac, and 2 cavity we have seen here. One is amniotic cavity and another one is chorionic cavity. For all these structures, it is known as week of 2. If we summarize the topics, that is, we have got here bilaminar germ disc formation, epiblast, hypoblast, trophoblast, primitive yolk sac, secondary yolk sac, amniotic cavity, chorionic cavity, Hoscher's membrane, extraembryonic mesoderm, extraembryonic somatopleuric mesoderm, and splachnopleuриc mesoderm, primary villi, connecting stalk. So these all the structures are formed or developed during the second week of development. Thank you all.